a great privilege for me to be here today as well, so thank you for having me. And just to sum up what Ramesh said, he's the pretty face, and when he's in the pub, I'm doing all the work. So
that's what happens if you're a senior consultant. That's what you do.
Okay, so you've seen a version of this slide a few times this morning, so I'm just going to quickly go through it. So we know that the minority of adult GIST are called so-called wild-type GIST. And if we look at the wild-type GIST, they can be further classified. And the first step, as you've heard this morning, is to determine whether the GIST have a functioning succinate dehydrogenase enzyme complex or not. And we usually do this using STHP immunohistochemistry or a staining test on the tumours. And we say if the tumour stains positive for STHP, then the STH complex is preserved. And if the tumour stains negative for the STHP protein, then the tumour has a non-functioning succinate dehydrogenase enzyme complex, or we call it STH deficient. And again, as we've heard this morning, the causes for STH deficiency in our wild-type GIST tumours include germline mutations in 4 genes, STHA, B, C, and D. But also we see patients who have this abnormality in their tumour where there's hypermethylation in the STHC gene, and this modifies the gene and affects expression of the gene in the tumour. So if we look at our UK cohort and look at the STH deficient GIST, we see by far the most common cause for STH deficiency is a mutation in the STHA gene. And I think this also holds true for the NIH clinic as well. And the second most common cause, again, as we've seen earlier this morning, is this STHC epi mutation. So we've also found that approximately 1/3 of our patients, when they present with their STH deficient GIST, will have a second primary tumour. And this holds true for patients who both have germline mutations in the succinate dehydrogenase genes, but also patients with STHC epi mutations. And the most common second primary tumour we see in this group are paragangliomas. And just to give you an example, you see in the top picture here, the red arrow, that's marking a carotid paraganglioma in a patient who presented with a metastatic GIST. And in the bottom, there's a red arrow pointing towards an abdominal paraganglioma in a patient who also presented with a GIST. And the image closest to me is a patient who presented with a pulmonary chondroma and an STH deficient GIST. So again, we've heard this morning that we really rely on our pathologists to help us differentiate between STH deficient and STH preserved GIST. And again, there's some characteristics that we've already seen this morning. So when our pathologist looks down the microscope at the STH preserved GIST, he tends to see these spindle-shaped cells. And again, as we've heard, when we do the immunohistochemistry test, the STHB protein is preserved. Whereas when we look down the microscope at our STH deficient GIST, we see an epithelioid morphology of the cells, or mixed epithelioid, and we see loss of the STHB protein. And again, as we've heard this morning, in our series, all of our STH deficient GISTs have arisen in the stomach, and we have had no case of a small bowel STH deficient GIST. So again, just to summarise our UK cohort, again, they tend to be very young in presentation. The tumour has always been in the stomach in our series. Histology tends to be epithelioid or mixed epithelioid. We unfortunately see high rates of metastases at presentation. STHA is by far the most common of the succinate dehydrogenase genes to be implicated in STH deficient GIST. And the most common variant we see in STHA is this truncating variant in arginine position 31. But again, as we've heard this morning, it's very important to remember that genetics is not always the answer for why these GISTs are STH deficient, and we see quite a significant proportion of our patients have an STHC epi mutation as a cause for STH deficiency. So just an example of how we go about diagnosing our patients. This is a busy slide with an algorithm of how we approach our patients. So again, molecular profiling happens to tell us whether the patients are GIST or conventional GIST or wild-type GIST. And if a patient is referred to as a wild-type GIST, the first step is to do STHB immunohistochemistry. If a patient is found to have an STH deficient GIST, the first test we do is germline genetic testing. As we've heard this morning, this is very important to inform ongoing surveillance and management for our patients, but also for family counselling. And these are the genes on our panel that we test for in the germline. If a patient is found to have a germline mutation in one of the STH genes, then they immediately come to my clinic, where we do surveillance every 2 years using MRI, looking at the skull base and neck for evidence of paragangliomas. And frequently these patients are having regular abdominal and pelvic imaging because of their GIST, and then they will get annual biochemistry to look for secretory pheochromocytomas or paragangliomas. If a patient has an STH deficient GIST but isn't found to have a germline mutation, we then go on to do testing for an STH-C epi mutation. And if a patient isn't found to have a germline mutation or an STH-C epi mutation, we then enter them into some research studies where we're doing some whole exome sequencing analysis to see if we can find other genes which might be causative. Okay, so I just quickly, and I'm aware that we are tight for time, I wanted to briefly mention some of the translational clinical studies that we have been involved with in Cambridge. So one of the issues which we face in the clinic is that some of the alterations in the succinate dehydrogenase genes may result in the tumours developing, but the alterations may not be so severe that they affect expression of the protein. So this means that for some patients, they may have an STH deficient GIST, but we're missing them by doing the routine immunohistochemistry. And that may be because the alteration is very mild, and it might affect the function of the enzyme, but may not affect the protein expression. So we wanted to identify other methods of picking up STH deficiency. So just to remind everybody, so the STH enzyme complex is made up of 4 proteins, and each of them are encoded by the 4 genes I've mentioned this morning. And if there's an alteration in any of the 4 genes, it means that this complex won't function properly. And if this enzyme doesn't function properly, then this metabolic cycle that you see here, which acts in every single cell, will not function effectively. And this metabolite succinate cannot be converted to fumarate if the enzyme complex does not function properly, and what we see is an accumulation of succinate. So we wanted to determine if measuring succinate accumulation could be used as a more sensitive way of picking up STH deficient tumours. So to do this, we've been taking fresh frozen tumour samples when available from our patients and looking at the metabolites. So I wish the picture was working, but oh, it is. I'm just pointing too far away. Okay, here. So these tumours at the top are tumours where we don't have an STH mutation, and these tumours at the bottom do have an STH mutation. And we see this peak here is succinate accumulating in these tumours, but not in the wild-type tumours. And when we've looked at the concentration of succinate, it's 15-fold higher in our STH mutated tumours compared to tumours that don't have an STH mutation. And we've been able to find a cutoff of 0.78 millimolar. That's a concentration of succinate, which tells us whether a tumour is or isn't STH deficient. So a tumour with a succinate concentration greater than 0.78 is suggestive of an STH deficient tumour. So we're doing this particularly for patients who may have STHB immunohistochemistry, which suggests preservation of the STHB protein, but who perhaps we suspect may actually have an STH deficient tumour. For example, if it's a young patient, a gastric tumour, we're doing this testing. And to take this one step further, we wanted to see whether we could look for succinate accumulation in tumours which have not been removed from the patient. For example, some of our patients, as we heard this morning, may not actually have surgery. They may just have a biopsy. And to do this, we used a technique called MRI spectroscopy. So a patient would have a normal MRI, but in the MRI are 2 coils, one which receives signal and one which transmits signal. And this enables us to look at metabolites within the tumours. So these are some examples. So both of these patients had a STH deficient GIST. Here we go. So both of these patients unfortunately presented with tumours in their liver, so liver metastases. So as you can see, we did an MRI spectroscopy in both of these patients, and here you see accumulation of succinate in both patients. And actually, in fact, both of these patients had an epi mutation. This is just some of their methylation analysis. This shows you how we can detect in vivo, while the patient still has the tumours in situ, we can detect STH deficiency by using this tool. And another example of how this could be used. So this is a patient who doesn't have an STH deficient GIST, but actually has an STH deficient paraganglioma. And this patient had an MRI spectroscopy performed on this tumour deposit. Here you can see succinate accumulation again. And this correlates with very high uptake of fluorodeoxyglucose on PET-CT. We did this scan before and after this patient was treated with 4 cycles of a special form of radio.